Well, first of all, a big thank you to ibiTea for inviting me to give this webinar, and thank you to those of you that are attending this afternoon. So over the next 30 minutes or so, I'm going to give you some an introduction into a protein called Willebrand Factor, give you some background to its structure and function and what it does, and some little snapshots of some of the research that we've been doing in our lab and why the kind of flow systems that we're using are really important for this research. So just to begin, it seems like quite a strange slide to start a presentation with. You've got on the left-hand side, you've got a picture of a like sort of normal home water drainage system. On the right-hand side, you've got a picture of the London Tube network. But actually, in many ways, both these images are very similar to this, which is the human vascular system. So, for example, if we have some kind of leak rather in one of our water pipes at home, we lose water. This can cause damage. Obviously, we run out of running water. On the other side, if we have a blockage on the London Tube network, this will bring the whole of London to a complete standstill. So in exactly the same way, we don't want to lose blood from our vascular system, like we don't want to lose water from our homes, but we don't want a blockage, which can lead to very negative effects, the same way that we don't want a blockage on the London Tube network. So our bodies are held in a very tight hemostatic balance, and hemostasis is incredibly important to keep that blood flow normal. So it's a very important process that prevents excessive blood loss, but at the same time prevents excessive blood clotting. Now, just in the interest of time, I'm going to give a very brief overview of how this process occurs, and believe me, this is a very brief overview. But essentially, following vessel injury, your body works very quickly to limit blood loss. For example, your vessel will undergo vasoconstriction, and then we have this process where we form what we call the primary platelet plug. And this is where platelets adhere to the site of vessel injury. And in the next slide, or the next few slides time, we're going to find out the role of Willebrand factor in this. But platelets will bind to the site of vessel injury. They will form an aggregate, they will form a loose platelet plug. And then we have this process which kicks in, which forms a fibrin mesh that will then stabilize that platelet plug. And the image on the bottom right of the screen here is just an image taken in our lab of what that fibrin mesh looks like. So any kind of vessel injury, our bodies will act to limit the blood loss, but then at the same time, will then work to make sure that this thrombus or this clot which is forming doesn't get too big. So where does Willebrand factor come into all of this? Well, VWF is a large multimeric plasma glycoprotein. In fact, it's one of the largest circulating proteins that we have. And within our vascular tree, it performs two major functions. It's the carrier molecule for factor VIII, but most importantly, it basically supports platelet adhesion to these damaged vessel sites under high shear stress. It's expressed within the endothelium, and it can range in size to a massive 40,000 kilodaltons, like I said, making it one of the largest circulating proteins. So we run these things called Willebrand factor multimer gels. They're specialized non-reducing gels, and this just gives you an idea of what the protein looks like. And I think the key take-home message from this is that we have these kind of series of multimers, the largest multimers at the top of the gel here, and it's the protein species that's here, these largest multimers, that are the most active in our vascular system. When it's synthesized within endothelial cells, it also forms these characteristic structures called viable Pollard bodies that are a massive hallmark of the endothelium. And these viable Pollard bodies contain these really ultra large VWF multimers. Now, when we teach this to medical students and to science students, we often describe VWF as being like a ball of string. And you can see it in this atomic force image here. This is actually an image of the protein, and it does very much look like a globular ball of string. And as we're going to find out during this talk, that if we expose this ball of string to certain surfaces or to shear forces, we unravel our ball of string, and this is highly important for its function. So this diagram here just gives you an idea of the structure. It's comprised of repeating domains. And there's three things I really want you to just keep in mind as I go through this talk. The first thing is that VWF contains an abnormal number of cysteine residues. So approximately 3.5% of the amino acids are cysteines. This is going to be a little bit important later. It also contains a lot of glycan chains. It's decorated with 16 N-linked and 10 O-linked sugar sites. And highly significantly, VWF is the only circulating protein which presents the ABO blood group sugars. So those same sugar structures that are present on your red cells that determine your blood group are also present on the VWF molecule. So how is the protein synthesized? Well, it's a large protein. It causes a lot of stress on the endothelium to synthesize it. In the ER, once a VWF monomer has been made, we have the kind of full-length monomer. It has the propeptide region attached here, which is comprised of the D1 and the D2 region. And within the ER, the signal peptide gets removed. We have various folding processes. There's lots of post-translational modifications, including the addition of those ending glycans, but significantly dimers form. And dimers are formed between adjacent cysteine knot domains. Now, once a dimer is formed, that's the cue for the protein to exit the ER. There isn't the physical space in the ER for this protein to really get any bigger. It then passes out of the ER, and in the Golgi body, what happens is that these dimers now become multimers. And multimerization involves disulfide bridges formed between adjacent D' and D3 domains, but it also involves the propeptide, and the propeptide is essential the multimerization process to occur. Once the multimer is fully formed, the propeptide is cleaved off and it remains non-covalently attached to VWF, and then the protein is either directed for storage within viable Palladio bodies in the endothelial cells or is released into the vascular system. Now, these electron micrographs were taken at Tim Springer's lab in America back in 2017. And what these images beautifully show is that this part of the molecule here, this kind of C-terminal bit, forms what we now refer to as the stem, with the rest of the molecule being referred to as the bouquet. So it's almost like a bunch of flowers. And what we understand is that in every dimer, this kind of C-terminus sort of folds itself up into this stem structure. And we now believe that the more closed the stem is, the less responsive the molecule is to shear forces, and i.e. it kind of follows that the more open the stem is, the more responsive the protein is to shear force. So to highlight how important this protein is, there are two things. The first thing is that there's a disease called von Willebrand's disease. VWD is the most common inherited bleeding complication worldwide, and approximately 1 to 2% of the world are affected. It's broken down into 3 subtypes. Type 3 is luckily the rarest, but it's also the worst subtype. Patients have no plasma VWF at all and experience very severe bleeding. Patients who have type 1 VWD produce a normal protein, but they in some cases don't make enough of it. So type 1 VWD is characterized by reduced plasma levels. And this can range from patients having very, very low levels and having a very serious bleeding type, to some patients going through their whole life and not realizing that they have VWD. Type 2 VWD is caused by functional defects in the von Willebrand factor protein. So there's some kind of change, which means that the protein cannot perform its function properly. It can't bind to platelets, or those large multimers that are essential for its function cannot form. What's also been recently recognized is there is now a 4th subtype, which is classified as low VWF. And actually, clinically, this poses a bit of a problem. Some patients with low VWF won't experience a bleeding phenotype, with other patients with low VWF can actually have a very serious bleeding phenotype. And this is something which actually many clinicians haven't recognized, but is now becoming very important. And these 2 images just really highlight how serious VWD can be. So this is just an image of someone who's posted this on Twitter, saying that welcome to life with von Willebrand's disease, where even a simple flu shot can make you bleed like mad. And patients will also experience very easy bruising. Now, on the flip side of the coin, so we've got von Willebrand's disease where patients bleed. It's also becoming apparent that von Willebrand factor plays quite an important role in cardiovascular disease. And we have evidence in animal models. So, for example, when the VWF knockout mouse was crossed with the LD-LR knockout, what was noticed was that not only does the VWF knockout form less plaques, it also forms smaller plaques as well. So, in some way, deficiency of VWF seems to protect against plaque formation. Likewise, in a stroke model, the VWF deficient mice are actually heavily protected against stroke. So, for example, this is an infarct formed in a wild-type mouse at day 1, and if you compare the size of the infarct formed in the VWF knockout mouse, you can see that it is much, much smaller. Likewise, in a DVT model, so a deep vein thrombosis model, absence of VWF or total absence of VWF leads to virtually no thrombus being formed. So certainly in animal models, we can protect against cardiovascular or any disease which involves a thrombus forming by absence of VWF. And it does actually appear that in man, this actually follows suit. Okay, so for example, there's now numerous studies. I've just got very few on this slide, but it is becoming quite clear that high VWF levels do actually represent a real risk factor for an increased risk of cardiovascular disease. So, for example, some work from our lab demonstrating that high VWF levels increase the risk of heart attack. High VWF levels increase the risk of first ischemic stroke. And likewise, patients that have VWD, so the patients that have these bleeding phenotype, have a greatly reduced risk of developing either venous or arterial thrombosis. We also have a disease called ATTP, which I'm going to come on to in the next slide, which is directly associated with accumulation of uncleaved VWF, and this leads to microclot formation and organ failure. So TTP, or thrombotic thrombocytopenic purpura, this is a result of those ultra-large VWF multimers, and this is due to either non-functional ADAMS13. So ADAMS13 is the enzyme that controls the multimeric size of VWF, or also antibodies directed against ADAMS13. So coming back to these gels that we can run, so this is what we would expect a normal person to have in their plasma. This is a kind of normal range of multimers. But what you can see in a patient with TTP, those larger multimers, not only are they increased, but they're much higher. Okay, so there's a big, massive increase in the ultra-large VWF species. Now, these patients develop platelet-rich thrombi, and the consumption of platelets due to excessive VWF binding the platelets leads to thrombocytopenia, and unfortunately, if untreated, episodes of TTP can be fatal. And just to keep things topical, we also live in an era dominated by COVID. Now, numerous studies have demonstrated extremely high levels in very severe COVID infection. This is just some data taken from a cohort of patients which we analysed during the first COVID wave back in 2020. And the real take-home message from this study is that compared to the controls here, which have a kind of normal range of plasma VWF, patients who were receiving treatment for various kidney-associated problems and were also infected with COVID and had very severe COVID infection, had plasma levels of VWF that we'd never ever seen before. So these are just unbelievably high levels. And what was apparent as well when we tracked patients over time, that during the initial course of infection, these plasma levels of VWF were very high. So this is just one patient who was monitored over 48 days. And it was really by the end of that 48-day period that the patient's plasma VWF levels had returned to anything which we could call normal. But during the initial phase of the infection, the plasma levels were very high. And as I'm sure that many people are aware, COVID-19 has been associated with a massive increased risk of thrombosis. So your plasma levels vary significantly, and there's many factors which affect your level of VWF. But I mentioned to you on my first slide about the ABO blood group, and it actually follows that your ABO blood group is a significant determinant of how much von Willebrand factor you have. And there's been numerous studies that have shown this, but perhaps still the best characterized is a large study back in 1987. And this demonstrated that individuals of blood group O have 20 to 30% less plasma VWF than individuals of the non-O blood groups. And routinely, it's seen that the plasma levels are lowest in group O and are highest in group AB. And it's also worth noting that actually individuals of blood group O seem to be more prone to bleeding problems, whereas individuals of blood group AB seem to be more prone to problems associated with thrombosis. So, okay, so what does this protein do? So we've got this big sticky protein that circulates like a border string and is highly responsive to shear stress. Well, as I said, it's the carrying molecule for procoagulant factor VIII. But the most important function, which we're going to focus on today, is it mediates platelet capture at sites of vessel damage. If we go back to our cartoon structure of the VWF protein, the two things that I'd like to take away from this, or well, three things really, is that first of all, VWF contains binding sites for sub-endothelial matrix proteins, principally collagen, which is located in the A3 region here. It also contains a binding site for platelet glycoprotein 1b alpha. So this is a protein present on the platelet surface, and this can directly interact with VWF. And it's also worth noting the adenosine cleavage site in the A2 region here. And like I said, the molecule is highly responsive to shear forces. So these are the range of shear forces which we encounter in our vascular system, and we currently take anything over 5,000 inverse seconds as being abnormal. As this image here shows, that the more shear force we apply to the molecule, and I'm going to show you some movies and images in a few slides time which confirm this, but the more shear force that we apply to the molecule, the greater the conformational change that takes place. And as these images here show, that at 1500 inverse seconds, our protein appears in this kind of border string, this kind of globular structure. But if we expose it to a shear force of 5000 inverse seconds, it will extend into like a kind of elongated piece of string. So how does the molecule function? Well, this is a slide that I often use for teaching students. But essentially, and just to note as well that nothing is drawn to scale on this. Obviously, the platelets would be much bigger than the protein itself. But the platelet binding site in the VWF molecule is essentially hidden. So platelets and VWF can happily circulate around. There's no exposure of the subendothelial matrix. There's nothing for VWF to bind to, and those platelet binding sites are hidden. So VWF and platelets can't bind and interact. However, following some kind of vascular injury, the endothelium becomes damaged, and this exposes the subendothelial matrix. Now, this then exposes a ligand for von Willebrand factor, so mostly collagen. So now this big border string has a collagen surface to bind to. When it binds to the collagen surface, it can undergo a conformational change, and this now essentially exposes the platelet binding site. Platelets can now come in, bind to the VWF molecule, and this can begin to form the primary platelet plug. Also, at the same time as well as exposure of the endothelial matrix, the endothelial cells that kind of surround the injury, they will be stimulated to release their stored VWF protein. So here we just have some control HUVECs. And on the right-hand side here, we have stimulated HUVECs, and what we can hopefully see is these big long strings, which big long strings of VWF itself. Now, I'm sure in other sessions, people have spoken to you about culturing HUVECs under flow, and we know that if we culture HUVECs statically, they form this kind of nice cobblestone layer. However, when we culture them under flow, they elongate in the direction of flow. But what we can do is if we stimulate these and we perfuse it over platelets, what we see is that the VWF protein gets released from the endothelium, and we see these long strings forming that represent platelets that are bound to the VWF protein. So likewise, we can just show it again in this movie here. And what you'll see at the end is a very clear platelet string. So these are endothelial cells, they're releasing their VWF contents. The VWF protein is becoming attached to the endothelium, and it's then able to capture platelets, and in this way contribute to the formation of the primary platelet plug. And this image here just shows a dual stain image where you can see the VWF fibers or VWF strings that are being decorated with platelets, rather like beads on string. But most of the assays that we do to look at VWF function in vitro, we're trying to mimic this setting where we have a damaged blood vessel. And these assays are actually relatively easy to do. So whole blood will be drawn from a donor, and we can either use it as whole blood, or we can process it to plasma-free blood, which is essentially red blood cells and platelets, and then we can spike recombinant or purified VWF back in. We use a dye to label the platelets, and then we just use a very simple syringe pump to perfuse fluid through our flow slides, and then we can use a microscope to image in real time the extent of platelet capture. Now, we routinely use the ibiTea 0.1 slides. These allow us to have high shear rates, but with a relatively low blood volume. And we typically use a mix of collagens type 1 and type 3. And we normally use something called diox-6 to label the platelets. So what you can see here is just a very simple assay. So we have our surface, we've perfused whole blood through this, and what you're seeing over time is the formation of these thrombi. And we can just zoom in a little bit on these, so they're quite intense. But these thrombi are formed by VWF binding to this surface and then subsequently capturing platelets. And then, of course, we can do all sorts of analysis on this to work out the surface coverage that we have with our platelets. Now, it's worth noting that everyone's blood is different. Now, routinely or in general, the more shear that you apply, the more platelet capture that you observe. In a typical assay with whole blood, this is what we would see. So 1500 inverse seconds, which I should say is kind of a fairly standard shear rate to use for assays like these. We have our basal level of platelet capture. A lower shear rate of 500 inverse seconds, we've got less platelet capture. If we increase the shear rate to 3000 and even higher to 5000, the extent of platelet capture has been increased, and we can show it here as well. So if you compare these kind of pseudo thrombi formed at 5000 inverse seconds to the ones formed at 1500 inverse seconds, you can see that these ones are much larger. However, recently we've been doing quite a lot of work just with healthy blood. So people are screened, we're making sure that they're not on any antiplatelet drugs. But specifically, what's been very, very interesting to note is that the variation in platelet capture has been quite profound. So with some people having very high levels of platelet capture, other people having very, very low levels. And what's also interesting to note as well is that the higher shear rates, so beginning to get to the point where blood flow isn't normal, some people seem very protected against thrombus formation at high shear rates, whereas other people seem to have very strong, have very strong thrombus, forming at high shear rates. So there's a real significant variation in platelet capture between individual donors, and these are very important factors to consider when you're planning experiments like this. So just to highlight what happens in type 3 VWD, and just to really show you that the system is highly dependent on the presence of the VWF protein. Once again, this is an IBD 6 0.1 slide. We have our collagen surface, and we're passing over whole blood. So again, you can see the kind of buildup of these platelet from Willebrand factor thrombi. If we perform exactly the same assay, but we take blood from a patient with type 3 VWD, they've got absolutely no plasma from Willebrand factor whatsoever. What you can see is that none of these thrombi form, and in fact, we don't even get individual platelets being able to attach to the surface. So this whole system is dependent on the presence of the VWF proteins, i.e. no VWF present, no platelet capture can occur. So we can look at ADAMS13 cleavage under flow as well. So remember I said that ADAMS13 is the enzyme which controls von Willebrand factor multimeric function. And normally, or before we had the flow assays, we would do this in a static assay, and we would use a multimer gel to assess cleavage. These assays would involve taking VWF, we would mix it with ADAMS13. We would have to had some kind of denaturant to cause a conformational change in the VWF protein to allow it to be cleaved. This would take several hours. And we could run this on a multimer gel and see that ADAMS13 was digesting these larger multimers into these smaller species. But obviously, these assays are not ideal, but they don't represent what occurs in vivo. But we can clearly see this if we take plasma-free blood, we can supplement it back with VWF plus or minus ADAMS13. And hopefully what you'll be able to appreciate by the end of these two movies, that if this is thrombi formed just with VWF alone, okay, so red blood cells, platelets, and von Willebrand factor, we can now compare it to thrombi formed with red blood cells, platelets, VWF, and where we've added in ADAMS13. So the presence of ADAMS13 doesn't ablate these thrombi from forming, but what it does is it controls the size of the thrombi, so they're less intense and they're a little bit smaller. We can obviously quantify this. What we've recently shown is that, remember that I said that blood group O individuals have less plasma VWF than the non-O blood groups. And we know from doing static assays that blood group O is cleaved faster than blood group AB, but we've recently shown this for the first time under shear stress. So in the absence of ADAMS13, blood group specific OVWF has a similar platelet capture profile to blood group AB VWF. What we can hopefully appreciate here is that individuals with blood group O, their von Willebrand factor protein is proteolized much faster, or visibly faster than individuals with blood group AB von Willebrand factor. We can also use the flow system to delve further into examining the interaction of the VWF-A1 with glycoprotein 1b alpha on the platelet surface. There's a couple of assays that we can do to look at this. So, for example, we can take our IBD slides, we can sparsely coat them with VWF protein, or we can take the IBD slides and we can saturate them. And we see two different types of platelet capture. What you're seeing in this movie here is we've got sparsely coated slides, we've got wash red blood cells and low numbers of platelets. And what you're seeing is what we call pause time. So this is where individual platelets are binding to individual von Willebrand factor molecules. They're pausing for a set period of time and then detaching, and there's nowhere else for them to really go to. Whereas if we have a saturated surface, what we can now see is that the platelets seem to translocate across the VWF protein. So essentially, the platelets are jumping from VWF or from each A1 onto the next one. So the bond between platelet and A1 is being rapidly made, but then rapidly broken, and these platelets are almost jumping across the VWF surface before coming to a standstill. So a number of years ago, we demonstrated that the glycosylation status of VWF is important for its platelet capture function. There's quite a lot on this slide, but I'll try and keep it as simple as possible. So essentially, the A1 region is the region which contains the binding site, and located between the A1 and the E3 region is a cluster of O-linked glycans. So we did some experiments where we took a small fragment of VWF just like this. We made it, and we made it with and without these O-linked sugar structures present. Now what you can see is when we have a wild type VWF fragment, so representing our normal protein, we saw an increase in platelet capture and platelet roll-in from low concentrations up until we saturate the surface. But with all these mutants that lack these O-linked glycans, it was only really when we saturated the surface that we saw platelets being captured. And when we zoomed in on this, what we noticed was that so these are overlay frames. What you can see is that in the wild type protein, there's a nice smooth transition between molecules. But if you look here, for example, you can see there's quite a lot of gaps. So again, this is representing a single platelet over several milliseconds rolling across VWF. And we think that these gaps are where there's just inaccessible von Willebrand factor molecules. So using a flow system like this, we've been able to demonstrate the cluster of O-linked glycans, or this kind of cluster of O-linked sugars between these two domains of VWF alter how platelets combine to the von Willebrand factor molecule. We've also been able to show that the higher shear that we have, the greater the conformational change that takes place, and with some quite profound effects. So 1500 ml seconds, so we in this assay, we've just taken a collagen surface. It's been perfused solely with VWF. So there's no platelets, there's no red blood cells. And then we've gone back and we've probed the protein with anti-VWF FITC antibodies. So at 1500 ml seconds, the collagen surface is decorated with VWF. But as we increase the shear rate, and now at this bottom here, we're getting up to very, very high pathological shear rates. What we start to see is this formation of these kind of like nets, or these big fibers that are orientated in the direction of flow. It's really worth noting that these sorts of shear rates are very, very fast, and we'll only see in conditions of when a blood vessel is very heavily occluded. So we can zoom in on these, and what you're seeing here is that if we really magnify in, we have a collagen surface we've perfused with VWF, and we stain for these really big, thick nets. So at very high pathological shear rates, the VWF protein forms this thick, dense, fibrous network. This is very, very prone to platelet capture, and it's also not very readily proteolized. So is this something which we can see in vivo? Well, yes, it is. And again, we can try to mimic this in vitro. So this is a custom-made flow channel which is designed to mimic a situation like this. So as we age, we obviously form plaques in our body, and these plaques will begin to occlude our blood vessels. And this occlusion, or this change in the kind of shear stress which is happening, has quite a profound effect on the VWF protein. So this ball of string, as it kind of reaches the apex of this occlusion, will make a transition from being a globular ball of protein into an elongated structure. And as it exits this kind of apex of this occlusion, it can then begin to refold itself back. So it kind of follows that at these very stenotic sites, this is where the shear rates are fastest, and this is where we're going to see most VWF function occurring. But we can mimic this in vitro. So this is just a flow slide like this. And what we clearly see is that at the part of the flow chamber where the blood flow is kind of relatively normal, a relatively normal speed, we see what we would consider to be relatively normal platelet thrombi forming. However, at this occlusive site here, what you can hopefully appreciate is we've got very big, dense build-up of platelets. There's a big, thick mass of Willebrand Factor platelets forming where the shear rate is at its highest. Now, we can show this as well using labeled VWF. So again, this is a custom in-house flow chamber. And in the movie you're about to see, what we have is we have whole blood, which is being perfused through a custom in-house flow chamber. And this has been spiked with FITC labeled VWF. I hope this movie plays for everyone. But essentially what you're seeing is big swathes of VWF protein, and being caught up in that big sticky mesh is lots of platelets. We even have some red blood cells. And this big sticky mess is highly resistant to Adams 13 cleavage. And what you should be able to see in a few seconds that part of this will also break off as well. Okay, and we can either eventually occlude this part of the vessel, or an embolic clump of platelets and VWF will break off. And if you imagine this occurring in vivo, this can become lodged somewhere in the microvasculature. Now, another way which we've optimized to examine this is a feature which occurs with VWF. If the shear rate is high enough, the molecule can spontaneously unfurl. And what we do is we take ibid 6 0.1 slides, we can coat them with purified VWF protein. We take plasma-free blood, so again, wash red blood cells and platelets, and we can spike in soluble VWF. Now, what we see is if we do this, if we don't have any soluble protein, we just have a series of platelets which will bind to the VWF surface. If we add back insoluble VWF, we get much more intensive platelet capture. Now, if we visualize this in real time, what essentially happens is that as the shear force increases, what you're beginning to see here, and again, I hope this is playing for everyone, is that as we're now getting to very, very high shear rates, the VWF molecule itself is unfolding in the soluble phase and is capturing platelets. And we're now seeing big swathes or big clumps of Willebrand Factor and platelets. And actually, interestingly, we don't even need any kind of thrombogenic surface for this process to occur. So it appears that the protein has this ability to self-aggregate or self-associate if the shear rate's high enough. So this is, again, this is a custom-made chamber. It has a central micropillar. And what we do is we can do in these assays, we can just simply perfuse labeled VWF. So there's nothing else going through. This is just labeled VWF protein. And what you're about to see in this movie here is this is just the protein which wraps itself around the pillar. But what happens is that over time, that big mass of protein is extended and more soluble VWF is bound or associated and extended itself out. So you can hopefully appreciate from these past few slides that this molecule really is dynamic and responds profoundly to shear forces. So hopefully what you can see is that there's to VWF, there's a good and a bad side. There's the good side that it captures platelets, we injure ourselves, it stops normal blood loss, and stops us from obviously bleeding. However, we have this unwanted side. We can have excessive unwanted platelet capture, very high shear rates. We can have these thick, dense fibers that form, and these can either occlude the vessel or these can break off and become lodged somewhere else. So a question at the moment that my lab is asking is it possible to separate the hemostatic side, so this good side, from this kind of pathological thrombosis, this negative side? One way that we've been looking at this, and this comes back to something else I mentioned at the beginning of the talk, is we've been looking at cysteine residues or the free thiols that are present in this part of the protein. So it was previously understood that all the cysteine residues that were present in VWF were paired up, so i.e. every cysteine took part in a disulfide bond. It's now, however, known that some of these cysteines can exist in a non-paired state. So it's a very simple way to show this. NEM and MPB are two molecules that bind to free thiols or to unpaired cysteines. If we take control VWF and we incubate it with MPB, we see a binding signal. If we pre-treat the VWF with NEM to block the free thiols, we lose that MPB binding signal. So this along with some mass spec, demonstrated that there was a series of residues that were present in the C terminus here and a couple present in D3, which can in some way exist in the unpaired state. So we decided to investigate how or what's the kind of function of this. So when we took control VWF our file blocked VWF, which we've called NEM VWF, what we could see was at 1500 inverse seconds, the difference between the control and the file blocks from Willebrand Factor in terms of platelet capture wasn't really that very significant. There was a slight reduction with the file block material, but there was nothing very significant. However, when we increased the shear rate up to 3000 inverse seconds, what became apparent was that the file blocked VWF was now beginning to lose function. And when we took the shear rate up to 5,000, as I said before, this is kind of beginning to go past the top end of the normal human range. What we saw was that whilst the control VWF could happily capture platelets, the NEM treated protein, so i.e. the file block protein, was virtually unable to capture platelets. So blocking VWF-free files was able to reduce VWF-mediated platelet capture in a shear-dependent manner. And likewise, this actually held true over a dynamic range. So this is going back to one of our in-house flow models again. And what you can see is that with the control protein, the parts of the slide where the blood flow is normal, we have normal platelet capture. It's beginning to get faster here. Again, platelet capture is normal, maybe slightly more. But when we get to this occlusive site, we're now seeing very strong platelet capture due to the molecule undergoing this very big conformational change and able to capture more platelets. However, with the file block material, again at the site which represents normal blood flow, the platelet capture is normal. But as we begin to increase or get to the part of the channel where the blood flow is increasing, we've got significantly less platelet capture. And at that very stenotic site, we've now managed to totally ablate VWF function. So likewise, we then went back and we did our assay again, where we just took purified VWF. We diffuse this over a surface. And what you can see again is that with the control VWF, we're still forming these big, long, thick fibers. So these big, long, thick fibers that are in the direction of flow. What you can hopefully see is that when we file block the protein, these fibers can no longer form. Okay, so not only do we have less fibers, they're very disjointed, they're not orientated in the direction of flow. So essentially, blocking those files can reduce the ability of VWF to form these thick fibers at pathological shear rates. So just to finish off, I'm not going to dwell too long on these last two slides, but essentially we've worked out using some atomic force models that this is due to the lifetime of the VWF collagen bond. So it's independent of its platelet capture function. What the key take-home message really from these two slides is that whilst the file blocked VWF can bind to its kind of substrate, that we can have some form of thromogenic surface, a collagen surface, the VWF can still bind. If those files are blocked, the bond lifetime is very, very short. So hence why this accounts for the reduction in platelet capture. And just to finish off, we did some in silico experiments to model what happens to these C-terminal modules that contain these unpaired files. And it appears that actually when there is a free file present, as one would expect, the molecule can be stretched slightly further. So our working hypothesis is that the free files can undergo a very local disulfide exchange. And this gives flexibility to the C-terminal region of VWF, and this allows the collagen-bound protein to withstand very high shear stresses. However, if you block these free files, you block this kind of flexibility, and it's almost like the molecule gets ripped off the collagen surface at very high shear rates. So just to leave you with three conclusions, hopefully you've taken away that effective expression or function leads to VWD. On the reverse side, elevated VWF levels are associated with an increased risk of cardiovascular disease. But I think most importantly, that actually flow-based assays are absolutely critical for investigating VWF function. And without some of the products that we've used from ibiTea, it would have been impossible to generate some of the data that we have done. So I'd just like to acknowledge the members of my group who have done some of this work. So also Professor Mike Laffan, who I've worked with for many, many years, who gave me the opportunity back in 2003 to begin at Imperial College. And I just want to leave you with this picture. I think in a time where the world seems to be going absolutely crazy, a number of weeks ago, we normally label our platelets green, but for the first time we labeled them red. And what we saw was this heart-shaped thrombus. So I would just like to leave you with that image. And thank you very much for listening.
Thank you, Tom, for this very interesting presentation. So I think I wasn't the only one who learned a lot. There are also some questions coming in, and you already get some feedback with emoticons. Maybe I'm going to start with the first one. Elizabeth is asking, is the individual variation in platelet capture at different flow rates related to plasma VWF concentrations?
No, that's actually a really good question. We thought it would be, but it's not. It's not at all. So we weren't expecting to see such a variation. All the healthy individuals were screened to make sure that they weren't taking any drugs that would affect platelet capture. When we went back to look at their VWF levels, there was no link between extensive platelet capture and von Willebrand factor. There was, at the highest shear rates, there definitely was. The 1500 inverse seconds, it definitely wasn't due to von Willebrand factor itself. What we haven't done yet is we think that it may be due to Adams 13. So it may be due to the ability of Adams 13 to control how big those kind of pseudothrombi are getting. So work which we're actually doing at the moment is working out what causes variation.
Okay, thank you very much. Next one by Piotr. Can I assume that the glycosylation is responsible for increasing the drag? So in essence, straightening the VWF from its globular to linear form like a kite or an anchor is doing.
That's a nice way of looking at it, actually. What has become apparent over the past year or so, some work from a chap called Renhal Lee has highlighted that the A1, the platelet binding region in the A1 is shielded by a short peptide region, which has been termed the AIM module. So it's the auto inhibitory module, and it appears that the only glycans that are on that peptide essentially stabilize that linker region. So what we hypothesized was that when those only sugars weren't present, that region was too flexible. So it's in some way the presence of those sugars stabilizes that region and then controls access to the GP1B alpha binding site.
Mm-hmm.
I hope that's answered the question.
Yeah, hope so too. A lot of people are thanking you for the great talk, by the way. And someone is asking if there's a treatment strategy for the VWF deficiency, or is this not treatable at all?
Yeah, no. So although VWD is very, very common, I mean, there's a very good chance that someone on this talk may have it. They may not even know about it. It is actually relatively easy to treat. We're very lucky in the third world that it's very in the first world that it is very easy to treat. Patients can be treated with concentrates, which are readily available. America has now got recombinant VWF, which is licensed and is actually very cheap to produce. In some cases, patients are given things like DDAVP to help the endothelium release the stores of VWF that are present. Also, tranexamic acid can also be used as well. So actually, luckily, although it does pose a problem, it can be treated. Unfortunately, the situation in the third world is less easier. Sometimes, obviously, countries which don't have kind of access to certain drugs, that's where it is more of a problem. I think it's also a slight problem in the fact that a lot of GPs sometimes are maybe not aware of it as well. So that can cause a problem, especially in young females who, for example, experience bleeding. They will go to their GP, and something which actually should be relatively easy to work out and then treat can maybe lead to other problems. So we have actually this year with our master students, we have actually been trying to make people more aware of diseases like this, which are actually relatively easy to diagnose and treat. But yeah, certainly in the first world, very, very easy. In the third world, I think slightly more tricky.
Yes, everything is, unfortunately.
Yeah.
Thanks. Someone is asking what temperature you run for the flow experiments.
Yeah, again,
They were quite short, right? Only 5 minutes as far
Yeah,
as I
they're 5 minutes. Interestingly, we haven't seen any difference between room temperature and 37. So we have set this up. We don't see any difference in platelet capture. We do, however, if we run the assays at lower temperatures, so if we drop below 10 degrees, platelet capture is virtually abolished.
Okay.
Yeah, which is interesting. So yeah, we don't see. I mean, they are very short. We kind of typically do a run for no more than sort of 5 minutes, 10 minutes maybe. But yeah, certainly room temperature is absolutely fine.
Okay, and 10 minutes is the maximum one usually does.
Yeah, I mean, we've never really had a reason to go beyond
Okay.
that time frame.
Just curious. Thanks. And someone has the same question as I do. What was the custom-made chamber made of?
Oh, right. So the custom-made chamber was made of PDMS. I think it's polydimethyl siloxane.
Thought so.
I think so. Something like that. Anyway.
Okay.
Yeah, I mean they are relative. They can be quite complicated to make. I mean, I have to admit we had a different lab who made those for us.
And how big were the channels?
Oh,
Let's say the small
big,
one for the stenosis model.
how big were channels? So the channels went from 200 microns wide down to about 55 microns wide.
Okay.
So yeah, very, very small.
Okay, thank you, and thanks for the question. Okay, did you look at possible dose responsiveness of NEM dosing on a ADAM-TS 813, activity and VWF multimers? In other words,
Right,
possible
yeah, we did. So we didn't see any effect of NEM on affecting VWF multimers up to the doses that we went up to. I have to admit, we haven't done anything to look at Adams 13 function, and yeah, in the flow assays, we did see a dose dependent effect of NEM on platelet capture function.
So maybe, Catherine, you can get in contact with Tom to discuss this in more detail, because you
Anyone's free
be
to
an
email.
expert as well. Okay. Okay, is there any difference on the platelet capture by VWF if the flow is steady or pulsatile?
Yeah, that's very interesting. We've done a few experiments with that. I can't say we've got any decent data to share on that, but that would definitely be very interesting to look at, because again, a lot of the assays that we do with a syringe pump, so it is a very, very, very steady flow. Yeah, absolutely.
Yeah, that's a very interesting question.
That's worth doing.
And the last one, I'm curious about local hemostasis agent using VWF disease. So probably also treatment again? Question about the treatment?
Yeah. I think again, just as what I said before, that
Right.
yeah, so most patients will either have some form of Willebrand Factor plasma therapy, so a concentrate recombinant protein, or in some cases a drug will be given to help the endothelium to kind of release any stores of protein that are there.
Thanks. I was quite surprised about the difference in the blood groups. So that was
very
interesting.
Yeah, it's a huge difference actually.
Yeah. So yeah, I'm not, as mentioned, not the only one who learned a lot.
Thank you.
Many were interested. Thank you very much for attending. I think everyone had a great time. And Tom, as I said before, as the audience said, great talk. Thank you very much for taking part.
Thank you. Thanks everyone for taking time to join me. Thank you.
Great. And then with this, I'm going to end the webinar. Thanks for participating. Have a good evening, everyone. Bye-bye.
All right, thanks, Peggy.
Bye, Tom.